Well, welcome everybody to the second GDPE Distinguished Visiting Ecologist Seminar of this semester. My name is Alan Knapp. I am the Senior Ecologist for the GDPE program. This is an awesome turnout. Raise your hand, would you, if you already know Sasha. Woo! Great, so that's awesome to have here. So I'm going to give an introduction to her for those of you who didn't raise your hand. And I want to, it might be a bit long, but I want to make sure I cover all of the accolades she deserves as our Distinguished Ecologist. You should know as well that this Distinguished Ecologist series actually predates GDPE. I think many of you know this already. I'll get the stats right. It's over 45 years old. 45 years we've had Distinguished Ecologists in this series come to CSU and make presentations like Sasha's going to do today. Eugene Odom was the first, right? The father of ecosystem ecology was our first Distinguished Ecologist. We had such luminaries as Dave Tillman, Phil Grime, Margaret Davis, Peter Vatusek, and Jane Belknap. Two I mentioned for you because she's actually co-authored with those two Distinguished Ecologists in the past. So Sasha fits into that mold quite well. So that brings me to my introduction of her. So Sasha is with the USGS Southwest Biological Science Center in Moab, Utah. She's been there for a number of years. We will not reveal how many. And I want to get to the standard biographical details for you before we get to the fun stuff. So she has got a bachelor's degree in organic chemistry from Colgate University. Pretty impressive, I would say. Any alums of Colgate here? Besides Sasha? Okay, so I can tell you anything I want to about Colgate University. I did a deep dive into this university because I used to have a graduate student in my lab who was from there as well, and I recall her telling me some interesting things about Colgate University. It used to be a football power. In 1932, it shared the national championship. 9-0, right? This is what Wikipedia taught me in 5 minutes of work. It also has a number of distinguished alums. They have a long list of distinguished alums from Colgate. Your name was not there yet, but I'm sure it will eventually be. But one of the names that was there was Ben Cohen. Anyone know who Ben Cohen is? He's the Ben and Jerry's. So he came from Colgate. I'm not sure if you overlapped or not. What was interesting about Ben is he did not graduate. He dropped out of Colgate University. Probably couldn't get through the organic chemistry program that you did. That's my guess. So after she graduated from Colgate, she spent 5 years, gap years, you might say. And she did a lot of interesting things, from skiing to going to South America to working for the USGS to working in the mountains. A lot of time was spent in Colorado, but she spent some time in Moab, and I think that's when she realized Moab is where, if she could, she would spend the rest of her working career. And it certainly worked out well for her. But after that 5-year gap, she then matriculated to the University of Colorado, where she did her PhD in biogeochemistry, so she kept the chemistry part. And in 2002 to 2008, she worked with Alan Townsend. She actually worked in the tropics for her PhD before moving back to Moab. So she's won many awards in her career. She's an ESA fellow, an AGU fellow, both. And in fact, I found this fascinating. She won the early career ESA fellow. She won the Simpson Medal in AGU for middle career scientists. And then she's also been a late career ESA fellow as well. So she sort of won awards throughout her career. I didn't see, you probably won one in kindergarten as well, I'm guessing, but it didn't show up on your CV, but I'm sure you did. So speaking of her CV, she's published over 200 papers, right? Last year alone, those papers were cited over, I have the number here, 3,000 times, right? She has 2 papers in journals I'm convinced no other distinguished ecologist has ever published in. The Tetrahedron Letters and the Journal of Organic Chemistry. She published 2 papers as an undergraduate in her chemistry program as well. So I'm confident even Eugene Odom could not doubt that. So her works are very impactful. Again, lots of citations. She's incredibly collaborative, which is why so many of you raised your hand in here today. She's a collaborative scientist in the best sense. She leads large groups, and I've been involved in those with her, and she supports others who lead large groups. And both of those roles are really, really important, and she does both really, really well. I'm fortunate enough to say that I have 4 papers that I appear on a long list of collaborators with Sasha, so I've ridden her coattails a bit during her career. So I've known Sasha since she was a graduate student. So when she was in Boulder, she actually came up to what we used to hold what we called mega lab meetings in, where 5 or 6 of the ecology labs would get together. Cindy, you remember that. Sasha came up for several of those from Boulder. And I knew then she was destined for greatness, right? That decision-making process alone, that she would leave Boulder and come up here and interact with the ecologists up here, I just knew there was greatness in her future. We did have free beer and food. That might have had something to do with it as a grad student as well. But nonetheless, I don't want to spend any more time. I want to turn the time over to Sasha. It's great to have her back here again. I will say this. I think it's always important when you give lots of accolades to people. You mentioned one thing they could work on as well. And since I've known Sasha forever, there is one thing she could work on, and that is curbing her enthusiasm for science. We'll find out today that she really struggles with that. So Sasha, you want to take it away?
Yes, thank you all. It's not the first time I've asked to have my enthusiasm curbed, but it's not my skill set, so we're not going to do it today. I'm so excited to be here. Wow, I just love coming to the Front Range. It's a homecoming for me. I love the amazing science that you're all doing. This is an epicenter of incredible, relevant dryland science and all kinds of science. It's absolutely magical. And I'm excited to think about some of the science that we've been doing together lately. And I gotta turn this on. And I'm going to tell you 4 stories, and heads up, that's too many. I know that I shouldn't, but I just couldn't stop myself. I really wanted to talk to you about 4 different things, but they're going to link together, and part of the link is that they're all going to have a hopeful message, a message about how when we learn about these ecosystems, it not only refines our understanding of the challenges they face, but the innovative solutions that we can bring to them to sustain ecosystems and communities, people, even in the face of change. I also note that I put here the number of slides that we're on and the total number of slides that we have to go. So if you need to zone out for a little bit, you can apportion your time and your mental capacity accordingly. I want to think together about some of the work that we've been doing on plant responses to climate change and the insight that that gives not only to how communities may respond in dryland systems, but how we could refine restoration approaches. I want to think about biological soil crusts and some of the ways we think those communities are responding to climatic change, but also information about innovative restoration approaches that could help sustain those communities. I want to think about pinyon juniper ecosystems in the face of increasing fire, and then a large-scale dryland, big science project that we hope those of you who aren't already involved will agree to be involved in. So those are the 4. Are you ready? Can we do it? Yeah. That was weak. So, why focus on drylands? I think if you ask a lot of people to close their eyes and picture a desert, they picture something like that. Blowing sand dunes, no vegetation, maybe there's a camel off in the distance on the hill. But we have perceived these ecosystems as wastelands, and that affects how we study them and how we make decisions about them. We don't often put the same resources into understanding these ecosystems, and we often manage them as if they don't have inherent value. And those things are changing. Drylands are having a moment right now. We're studying them in new ways. We're recognizing their global importance in new ways. They are our planet's largest land biome. They make up 42% or more of the Earth's land system. They make up over 1/3 of our country's land system. 1 in every 3 people lives in a dryland. They have 35% of the human population. They produce 60% of our food, which is a number that surprised me. And they play critical roles in Earth's carbon cycle and in regulating our climate. They determine the trend and the interannual variability in atmospheric CO2 and in the terrestrial carbon sink. So the uncertainties that we have about what drives their carbon cycle, about quantifying that carbon cycle, means uncertainty for all parts of Earth's carbon cycle. So it really hinders our ability to model and forecast what we can expect from the future. So they're really important ecosystems. They're also places that are changing fast. Here on the left-hand side, you can see the rate of warming for the last 100 years or so across every county in the United States. I'm just showing you the western US here. This bloodbath here is Moab, Utah, where I live. Grand County is warming the second most of any county in the United States. We see lots of other drylands in the US warming at very high, above average rates, including areas in Colorado. And it's not just the United States. Here we have Australia, and that purple bruised color in the middle is a new temperature that Australia's Bureau of Meteorology had to add to the temperature scale because the past one didn't go hot enough for the hotter temperatures that they're experiencing now. This bruise is in a dryland. So we have these already hot places getting hotter. And in fact, most of this warming is in a dryland because most of Australia is a dryland. And so we see these changing temperatures in ways that are worrisome. We also see dry places getting drier. Here we have the western US, and during the growing season, I've never gone to the drought monitor and not been able to pull up a map of the western US that has extreme drought and exceptional drought in drylands. We are in the worst drought in the Southwest in at least 1,200 years, and so we know these are dry places already very limited by water that are getting drier. And globally, it's not just drylands, but lots of drylands are experiencing aridification, both because of increased frequency and intensity of drought and because higher temperatures can mean drier systems even without a change to precipitation regimes. We also see effects of things like changing atmospheric CO2. This work by your own Dave. Obviously, you don't have many pictures on the web. I had to surcharge for this one. This was cool. So, and here we see this amazing face experiment that lots of the people in this room were a part of, where we have the normal temperature, normal CO2, elevated temperature, normal CO2, elevated CO2, normal temperature, elevated CO2, and warming. The warming did have effects on aspects of forage amount and quality, but you can see here the huge reduction in nitrogen in forage that happened with both elevated CO2 treatments, regardless of warming, with big implications for forage quality for wildlife and livestock. So these changes that are happening in dryland systems could really feed back to affect a lot of different kinds of organisms. And indeed, we do see that here, work by Craig Allen, formerly of the USGS Fort Office, stationed in New Mexico, showing massive pinyon mortality in New Mexico. So all of those brown trees are dead pinyon trees that died because of global change type drought, which just means drought that co-occurred with high temperatures. Now juniper trees are dying. And Craig and others he works with have shown this forest mortality in drylands across the world. So not only are the climates changing for these ecosystems, the organisms are responding to those changes. We're seeing threshold type responses, big die-off events of things like trees here. And that's worrisome. But luckily we have ecology to help us understand these systems. I went down such a rabbit hole trying to create the coolest ecology superhero, and then I tried it on ChatGPT and things got so weird. And so this is just something that I pulled off the web and then stuck ecology over parts I didn't want you to be scandalized by. And so, but ecology really gives us so many exciting tools for understanding how ecosystems are changing, why they're changing, and through that understanding, what can we do? What decisions can we make for ecosystems to sustain them and their functions moving forward into the future? So the first story that I want to tell you is on the Colorado Plateau. Here you can see the Colorado Plateau is my favorite desert, where we have a warming and altered precipitation experiment. There are two sites that are part of this experiment. The first one that you see here is relatively shallow soils with lots of good biological soil crust. The picture isn't showing it, it's a little pixelated, but all of this brown scorched earth soil is biocrust. We also have a deeper soil that's poorly crusted because of historical overgrazing. We have two temperature regimes, 2 and 4 degrees Celsius above ambient, so the infrared lamps track ambient temperatures to turn the lamps on or off so that each plot is the correct temperature above ambient. They work very well most of the time. and then two precipitation regimes, one where we had high frequency, small precipitation events, just little rain bursts happening on the plants and soils, and the other where we had larger, less frequent events to see how added water could interact with temperature to reduce or even eliminate some of those warming effects or not. And I'm not going to show you all of the data from all of the treatments, but some of them. Here we see photosynthesis of the C3 grass Achnatharum hymeneoides, Indian rice grass, which is a super important grass for us for livestock and wildlife on the Colorado Plateau. And what you can see here is that early in the growing season, warming reduced photosynthesis of this grass for both the normal precipitation year and the drought year. When we look at the C4 grass, Chloraphis jamesii, Guyetta grass, we see that warming did not have negative effects on photosynthesis except late in the season in the drought year. And so there's a couple of things that I want you to take home from these figures. The first is that whenever warming had an effect on photosynthesis, it was a reduction. So these plants were reduced in their ability to get food through photosynthesis by the warming treatment. The second is that these two different grasses that live in close proximity, one of the things that I think is so cool about drylands is that you can have C3, C4, CAM plants all living in a meter squared area. We have a lot of diversity at that level to see. So these grasses are living intermingled with one another, but the timing of the temperature effects on photosynthesis and the mechanisms are different. Data that I don't have time to show you show that this is really a drying effect, that the warming is reducing moisture for this plant early in the growing season, and that's reducing photosynthesis. Here, this seems to be a temperature effect. The plant is closing its stomates because of the water limitation of the drought year, and then the heat that it's not able to cool itself, that photosynthesis is reducing. So different ways that temperature is affecting these two plants that live together. I really like experimental data because you can push the system and be super impatient, but you can also use long-term data like Seth Munson, who got his degree here at CSU, did using the National Park Service inventory and monitoring, amazing data set of plants across time to also show a decline in C3 perennial grasses at many sites across the Colorado Plateau. So together we see the mechanisms, maybe reduced ability to get carbon, and the outcome could be reduced cover of this important type of grass in warmer years. Daniel Winkler also looked at Indian rice grass cover in our plots and found, just as you'd expect, there was a reduction in the cover of that plant in the warmed plots. And when he looked at photosynthesis 6 years after Tim Wharton did, he saw that photosynthesis was still reduced early in the growing season with warming. And here, looking at the relationship between photosynthesis and the temperature of the plant, we don't see any acclimation of photosynthesis. We don't see the plants moving their temperature optima of photosynthesis to warmer temperatures, which is part of why they're probably photosynthesizing less. They're not able to acclimate to that warming, so we're seeing them respond with reduced photosynthesis when the temperatures go past that optimum. But the plants that were living on the plot, so we had plants die, reduced cover, but the plants who maintained, who were there, were giants. They were monster plants, the biggest, I still call them stiger, the biggest Achnatharum hymeneoides that you've ever seen. And so we see this lots more biomass combs, lots more reproduction, which is data that I'm not showing you. And it looks like phenology plays a role here. The photosynthesis is not higher in these giant plants, but they have a longer time to photosynthesize. Their growing season is earlier with the warming, and it ends earlier as well, but it starts more early than it ends early. Is that correct English, I think? That makes sense, though. They have a longer growing season. And so we see these giant plants, and that gives a lot of hope for thinking about why. Why are those plants monsters? So we work closely with the National Park Service on questions of Indian ricegrass because of the ecological and cultural importance. And they're being told by some scientists that they might have to give up on Indian ricegrass, that it's not going to be able to be maintained in the hotter future. They're seeing it decline in their own inventory and monitoring data. But the monster plants in the warmed plots are plants that were born in those plots. So this experiment has been going since 2005. So there's a few different reasons of why those plants could be monster plants. One is reduced intraspecific competition. There's fewer of them, there's less cover, they're not competing as strongly for resources. A second could be maternal effects, that the mom plants who made seeds that became those plants prepared those seeds in a way that made the plants better adapted to warming. Or a third could be genetic properties, that there was a genetic selection that occurred, that plants that are just better adapted to warming. Whatever the answer, this is a restoration opportunity. So we took seeds from the control in the warmed plots and then planted them in separate control and warmed plots. And these are data hot off the press, so we haven't done much with them yet. But the suggestion is that the control seeds made seedlings that were less able to survive the warmed condition than the warmed seeds. So if this is true, this is restoration awesomeness, because it means that managers could find certain individuals or communities or could treat plants to grow seeds in a certain way that would create Indian ricegrass that is more capable of sustaining a warmer world on the Colorado Plateau and perhaps elsewhere, not have to give up on this species. So warming had only negative effects when there were effects on photosynthesis, cover. I didn't show you the data, but also reproduction. There were big differences in how plants responded to warming across those different species. The Indian ricegrass that made it, there were fewer that were there, but they were monsters. They were huge. And then those warmed plots made seeds that seemed better adapted, made seeds that made plants that seemed better adapted for a warmer world. Okay, story 2. We also have biological soil crusts. Biological soil crusts are a photosynthetic community of mosses and lichens and cyanobacteria that live on soil anywhere soil can see the sun, anywhere there's direct access to the sun. They can represent more diversity in terms of species richness at many of our dryland sites than the vascular plants, and they can play lots of important roles. Biocrusts are estimated to cover 12% of Earth's land surface, which is a ton. That's like almost twice as much as tropical rainforests. These are not boutique communities that we only love because they're super cute and beautiful, though they are super cute and beautiful. They are globally relevant communities that we have a very poor understanding of. But the data that we do have show that they're really important in soil stabilization. In the picture on the right, I can see this everywhere. Okay, in the picture on the right, you can see the cyanobacterial tendrils that are holding on to sand, that mineral soil. They produce this sticky exopolysaccharide glue that binds the soil together. They are phenomenal soil stabilizers. Of course, in the Southwest, we have big concerns about dust. Human health concerns, ecosystem concerns, loss of fertility concerns. It can be apocalyptic. It can rain mud in Moab. And so we want that soil to stay in place, and biocrusts play big roles in keeping that soil in place. They can also play big roles on hydrology of a system. On the left-hand side are data from meta-analysis, a global meta-analysis that showed, like I already told you, there was lots of reduced sediment production, reduced erosion in soils that had biocrusts compared to soils that didn't. It's this comparison. But we could also see 14% more soil moisture in soils that had biocrusts. For dryland systems, that's a huge amount of moisture. So they're playing these roles in hydrology. On the right, you're looking down at 6 pots that all had the same homogenized soil added, and then half of them had biocrust added, and a seed was planted in each. And you don't need a lot of fancy statistics to be able to tell that the biocrust had huge positive effects on the growth of that plant. When we looked at the soil after the experiment, we saw more nitrogen in that soil with biocrust. So the water part can play a role, but the fertility part too. Your own Carrie Havrilla has done amazing work linking biocrust to plant community success and improving our understanding of how these organisms interact with one another, how they may sustain or compete with each other. So here, going back to that climate manipulation experiment I was telling you about, you can see the cover of the moss Centrichia cananervis on the y-axis. And you can see here that it makes up a lot of cover of these sites, but with the altered precipitation experiment, where we increased the frequency of the small bursts of rainfall, we saw dramatic mortality of that moss. And that really confused me. I would have thought adding water to an organism in a desert would always result in a positive effect, but this killed the moss, and it was brutal. Doing these watering treatments, you're out there, you can like hear them, they're like, no, not again. And so then the question is why? Why does that moss die? And so here, looking at this work, we took these mosses back to the laboratory and then wet them up. It's time since wetting on the x-axis, and then if it's a positive number, the moss lost more carbon via respiration than it gained via photosynthesis, and if it's a negative number, it gained more. So when we added more carbon, 5 millimeter, more precipitation, 5 millimeter rainfall event, we can see at first there's a net loss of carbon. The moss is getting ready, it's opening its little leaves, it's preparing its photosynthetic machinery, it's repairing DNA, but then quickly it's taking up more carbon than it's giving off. With the small precipitation event, we see that there's never negative net uptake of carbon dioxide. They always lose more carbon than they gain. And so carbon starvation is one of the ways that this could be affecting these biocrust communities. Biocrusts are optimists. They don't have stomates that they use for gas exchange. They don't say, is this a big enough precipitation event for me to be active? They just go for it every time, which in past climates has been a really good evolutionary advantage. But now in a drier, warmer world, these little rainfall events have them open up. Oh, here, I'll show you. Have them open up. They spend carbon to prepare to take up more carbon via photosynthesis, but they're dry before they have the opportunity to take that carbon up. And I think this has really interesting links to the forest mortality that I was telling you about before, this carbon starvation mechanism for photosynthetic organisms dying in drylands. There, the trees are keeping their stomates closed in order to try to avoid drought, but that means they're not getting enough carbon to maintain themselves. So you can see these tiny little organisms, the awns, the little hairs on the ends of their leaves that they use to capture water. They're optimistic, and then we kill them. And so what we also see here with the moss cover is that the warming too had a negative effect on moss, but that effect took much longer to show itself. And we're still trying to work out the mechanisms of that mortality. In 2012, we stopped the watering treatment, because how long does something have to stay dead before you stop watering it to kill it more? And we saw good recovery in this blue line here. There aren't that many studies of standard biocrust recovery times, but this is much faster than we expected from some of that other work. So that's hopeful. We saw this recovery. But the warming treatments continued, and we saw that recovery eliminated when those biocrusts were attempting to recover after the climate precipitation nightmare ended. The warming meant that they didn't come back, suggesting problems for restoration. This is what that looks like. You're looking down here, you can see those beautiful biocrust communities, and here you can see, it looks like I made a crust angel here, but this isn't a physical disturbance. This is a climate-induced change to the biocrust community composition where we lost those mosses and lichens. And this has lots of big consequences for the system. One is the change in albedo, the change in energy exchange with the atmosphere. Here we have the control plots, and then these are the plots, the watered plots and the watered and warmed plots, where we see increased albedo, increased reflectance of energy back to the atmosphere. And for fun, we did a calculation where we said, okay, we have this map, this biocrust map. If we pretend that in terms of albedo, all biocrust looked like our control plots, and then we changed that so that all biocrust looked like our watered plots, that was change in albedo, we would have seen a net cooling effect on Earth larger than all anthropogenic radiative forcing of warming. And so I'm not telling you that that's the truth, that's a ridiculous calculation, but the scale of that suggests that the fact that we're not considering changes to biocrust communities in our Earth system models of ways that Earth changes, climate changes to systems could feed back to climate is a problem. So we're working with the Department of Energy now to include biocrusts into Earth system models so that for its carbon contributions and its energy balance contributions, we can better understand that. Here's a figure that I made and have hated ever since, where I was trying to show how changes to the biocrust community that occurred with the climate perturbation, the watering, affected the nitrogen cycle. And so it's basically the control plot nitrogen values divided by the watered plot nitrogen values. And so you can see here we saw large increases in nitrate with the moss mortality in the watered plots, and in nitrate and in nitrification rates. And that change in the nitrogen economy to more of a nitrate economy than ammonium economy could have as big a change on the ecosystem as a change in the total amount of nitrogen, because different organisms have preferences for different types of nitrogen, and because different kinds of nitrogen are more likely to leave the system through leaching in gaseous form. So big changes to the nitrogen there. And then here too, with the death of the biocrust, the climate-induced death, we see more losses of CO2 to the atmosphere from these high temporal resolution measurements of CO2. And that makes sense. We have lost these dominant photosynthetic organisms, and now it's much more of a source to the atmosphere. And so we've known for a long time that different kinds of land use affect biocrust communities, and these data show that climate could be having significant effects on biocrust too. And so that brings up the question, can we sustain these communities? And more than ever, land managers in the West ask me about how to support their biocrust communities. It's become more and more known that these are vital communities in the system. So how can we support them even in the face of this change? And as I said, we don't have enough standardized data of recovery times, but the data that we do have times hundreds of years, even 1,200 years. And this is not an easy to use value for managers. If you say, oh, I have some disturbed biocrust, what would you recommend? And I'm like, give it 1,200 years and you should be fine. So how can we increase the rate of biocrust recovery through action, through restoration activity? So one of the ways we started, work led by Anita Antonika and Matt Bowker, Northern Arizona University, was to start growing biocrust as restoration treatments in the greenhouse. And so these amazing systems that they set up to be able to grow biocrust, and you can see here, 1 month, 2 months, 3 months, 4 months, we were able to grow late successional biocrust, mosses and lichens in not 1,200 years, but 4 months. And so then we could use that for restoration materials. So like, send the Nobel Prize, we're totally ready, we solved the problem. But then everywhere we put those greenhouse grown biocrusts, they died. Great Basin died, Colorado Plateau died, Mojave died, Chihuahua died. So it wasn't working in the way that we intended. So we're trying a number of different things. One of them is the idea that we made life too easy in the greenhouse. We need the biocrust to be tougher when they go out. So we're growing them in these outdoor nurseries. At this time, this was the world's largest outdoor biocrust nursery, which is easy when you're the world's only outdoor bio. This sounds cooler the other way. And so growing them. We also were thinking about how to put biocrusts out with their community. I heard a number of you students today talking about island restoration, thinking about putting plants out together as a way to sustain each other. This is the same idea. So we grew the biocrust communities on biodegradable weed cloth and put that out. Often when biocrusts are put out for restoration, they're sprinkled across the landscape. So it takes advantage of the fact that biocrusts are totipotent, which means you can cut them up into tiny pieces and each of those pieces could survive and propagate. But it might be that they need that community to survive. Yes, you can break them up, but maybe if you put them out with the friends that they decided to grow near in an intact community, you can get them to survive. And we're seeing wild success with this. It does mean that you're restoring a much smaller area. You can't send these carpets out from space or from airplanes. Space would be cool. From airplanes, the way we do seeds, the way we could do broken up pieces of biocrust. But the biocrust that we do put out as an intact community is showing fantastic success. The third is that we're growing biocrusts from hotter deserts as well as from local deserts to say maybe we need biocrusts from places that are already the temperatures that we expect for the future. And this doesn't come without risk, right? This is assisted migration, and so there's pros and cons. We're doing the science not because we think it's the way to go, but because we think we need data in order to make the decision on what is the way to go. And so we don't yet see the idea that a lot of people think the Colorado Plateau is turning into the Mojave climatologically in terms of the plants. So we collected plants from the Mojave and the Sonoran, but we didn't see increased success of those communities yet, though that could play out in other ways. Okay, so biocrusts are awesome, and we should all be thinking about them in drylands all of the time. Also, they show inherent resilience to climate disturbance in some instances, but huge vulnerability in others. And the idea that warming could inhibit their recovery plays into concepts of degradation of drylands that need to be considered in lots of different frameworks. The consequences of those climate-induced changes to biocrust communities are real, but biocrust restoration approaches can consider those changes, come up with innovative attempts to try to address that change. I also want to tell you about CrustNet. This is something that started as Jill Barron and Marty Goldhaber started the USGS John Wesley Powell Center, which is basically the coolest thing ever, a science synthesis center. We got together and came up with the idea for CrustNet, and after like 82 tries, NSF funded it, and this is being led by Sierra Jack Google, who did her PhD at University of Colorado Boulder and is now the lead of this. So CrustNet is a global network to assess biocrusts, to assess diversity function relationships. There's different tiers of involvement, but also to do drought experiments and disturbance experiments to try to better understand what biocrusts do we have where across the globe. How are they responding to change? So we would love anyone to be involved. There's lots of different ways to be involved. Please consider doing so. You can reach out to me if my cool QR code doesn't work. So the third story, thinking about fire. In many ways, it feels like we're living in the Pyrocene, and the Front Range feels that in real big ways. And so we see much more frequency and intensity of fire in these ecosystems. In part, that has to do with the increase in exotic annual grasses that fill the interspaces between plants and drylands, allow for the carrying of fire much more easily. And in part, that has to do with aridification and the drying of fuels. So here in the yellow, you see for western US, the expectations of what wildfires would be in the absence of climate change, and this much more wildfire occurring because of climate change. Here we see the aridity, the dryness of fuel versus the fire, the burned area. In these drier later years, we see more dryness of fuel, more fire. And so that makes sense. We're seeing changes to fire regimes because we have a drier world, because we have more fuel filling that inner space. So this is a study that we did with the National Park Service, so Spector, she's now with the U.S. Forest Service, in Mesa Verde National Park, and with the Ute Mountain Ute Tribe and Ute Mountain Ute land south of the park. And these managers came to us to say they were concerned because they weren't seeing any pinyon juniper regeneration in their park and on their tribal land following fire in a time that they expected it to be. So I don't expect you to be able to see these words, but each of these polygons is a burn. There's more that have happened since this map was made. So basically, pinyon juniper used to be a dominant ecosystem type in Mesa Verde National Park and on Ute Mountain Ute land, and now it's a tiny proportion of ecosystem type because of the incredible amount of fire that's happening on these landscapes. And you can see the consequences. You know, they had a lot of these old growth pinyon juniper ecosystems, beautiful biocrust, lots of rare plant species, and now they have these cheatgrass nightmares with dead burned plants and monocultures of cheatgrass, which increases the likelihood of subsequent fire. This science, the managers came to us and asked for it, and we did it with lots of partners, which is a super fun way to do science. It was funded by the USGS North Central Climate Adaptation Science Center, which we have here on the Front Range. And it's a science center that focuses not only on science that can inform management and decisions, but science that's co-created with those managers, which greatly increases the chance that that information will be useful, will be accessible, will be used by those managers. So a very cool way to do science. And this is something that other Fort Collins USGS scientists and I are working on, Lydia and Emma and Sarah, on a training module for USGS scientists in how to conduct co-produced science. And I think about my own early self and the really weird ways that I went about trying to create relationships with land managers to try to better understand their needs. This is an attempt to create useful information, accessible information about how to understand what managers have on their plate, about the ways that they make decisions, not only with science, but with public opinion, with the financial and personnel resources they have, following the law. We do hope to make this publicly available in general, and so it would be something that could be virtually available, but also to have in-person trainings for USGS scientists to improve our capacity, our ability to meet our partners' important needs. So after that aside, back to the pinyon juniper ecosystem. Pinyon juniper systems, we often take them for granted, kind of like, oh, I'm camping out in this PJ and like, what am I even doing here? It's hot. But they're actually a really important dominant ecosystem type in the western US, or one of the most prevalent forest types. They have enormous ecological and cultural benefits and resources, including species that rely wholly on them in order to survive. And I really like thinking, I won't go too far into this, but we really don't like pinyon juniper in some places. We decide to remove it in many parts of the Great Basin. We love it in other places and are worried that it's not coming back. And I think thinking about how we perceive ecosystem value, how there can be different ways that ecosystems are or aren't useful, they're changing differently across the landscape, is really cool, really interesting. But in this place in Mesa Verde and on Ute Mountain Ute land, they love their PJ, they're losing their PJ. So there were two parts to this experiment. We compared unburned, unthinned pinyon juniper that we thought would have the coolest and wettest conditions, an abundance of the rare plant species that managers were concerned about, lots of the mycorrhizal fungal symbionts that we know pinyon and juniper need to survive, high soil stability and fertility. We thought the opposite would be true, in the burned area. And because we were working with the managers, it became clear how concerned they were about what thinning might be doing to their ecosystem. And so they asked if we could add a thinning treatment, and so we said, yeah, that sounds awesome, and we thought that many of these things we were planning to explore and measure would fall in the intermediary between intact and burned for the thin. We also planned a manipulation experiment to plant pinyon and juniper seedlings in the canopy of shrubs, with and without their mycorrhizal partners. And originally I had what I thought was the super cool idea that we would build these little structures to try to alter microclimate without having the fertility differences of the shrubs, so we could tease apart whether any shrub effect was because of cooler and wetter conditions versus more fertile conditions. But the managers rightly were like, well, we're never gonna do that. And so could we use some of the dead trees that we have out instead? And I was like, oh yeah, that sounds good. So here we can see sapling count in the intact forests for pinyon and juniper. And what I want you to see here is that we do have lots of recruitment in the intact forest. We do see this for seedlings as well. There's seedlings and saplings that are coming up in the old growth pinyon and juniper. I'm showing you the fire data here, but you can't tell because we found 0 pinyon and juniper seedlings or saplings in the burned area. It doesn't mean that they couldn't come back. It's been maybe 25 years at the time that we did this study, but the managers who know this ecosystem well were concerned that they would expect to see them coming back. When we looked at the effect of thinning, we had 2 types of thinning. We had a more mild type of, and so just to say, the thinning is being done in these ecosystems to reduce the chance of stand replacing fire. So the managers are coming in to remove trees to reduce the chance that fire is going to move across this landscape. When that thinning happened in a more mild way, we saw no significant reduction of pinyon, juniper, seedlings, or saplings compared to the intact forest. But when it happened in a more extensive way, taking many more trees, chipping those trees, and spreading those chips across the landscape, we did see reduced pinyon and juniper establishment, suggesting a sweet spot of thinning, where you could still remove trees to reduce the chance of stand replacing fire, but you could reduce your unintended consequences on the new pinyon and juniper organisms coming up. We also looked at biological soil crusts in the intact, thinned, and burned sites for the lighter, more milder thinning, and for the more intense thinning. We saw that fire rocked the biocrusts regardless of where they were. It was a big negative effect on those biocrusts, but that this lighter thinning had no significant effect on biocrusts, where there was a larger reduction in biocrust with that heavier thinning than the fire had. So huge effects of thinning treatment. Again, suggesting there are ways to perform the thinning that can allow these other members of the community to be sustained. The second part of the experiment was the planting done by Jillian Trimber, Northern Arizona University. So she grew hundreds of the cutest baby pinyon and juniper plants you've ever seen, and then planted them under shrubs, out in the open, under the downed plants, all with and without their fungal symbionts to try to tell the difference. And these are her preliminary results, but what she found was strong positive effect of survival under the shrub, under this kind of nurse plant framework. It makes sense. And a significant but milder positive effect of the mycorrhizae. So having mycorrhizae did matter, and that's cool. That's something that we can add to seeds and to seedlings. And where you put those seedlings had huge effect on the landscape. So we didn't see pinyon and juniper coming back in the burned areas as the managers feared, and we did see the fire crushing the biocrust communities. That thinning method had a huge effect on the outcome and on the unintended consequences, which is awesome. That means we can plan our management around thinning in a way that gets us our reduced fire outcomes but doesn't affect these other systems, that fungal partners and nurse plants did significantly increase PJ survival, that they were planting, and that the restoration treatments we're doing are things that these managers are now doing on the landscape in these places in order to try to facilitate the regeneration of the plants. You did 3 of the 4 stories, and now it's the final story. I want to talk about NASA Terrestrial Ecology Field Campaign that's being led by your own Dennis Ojima, Karen Prentice, who is the National Science Advisor of the Bureau of Land Management, who's here in Fort Collins, Bill Smith, who did his degree in Fort Collins. I basically only work with people who are in or were in the Front Range, as well as a lot of other people in this room and outside of this room who are planning this large campaign that we hope will sustain dryland science for the research community and for our manager partners. So the NASA Terrestrial Ecology Field Campaigns are meant to ask big science questions, to help enable better interpretation of our space-based assessments of ecosystems, to foster collaborative interactions, to provide opportunities for our very important next generation of scientists, and to leave a legacy of data. And the idea is that they cross scales. You use space-based data, you use airplane, you use drone, you use ground-based data, because these complicated systems and the way that they're responding to change won't allow you to use only one method or one scientific discipline and get the answer that you need. The NASA Terrestrial Ecology Field Campaign that's currently happening is above. It's ending the Arctic and Boreal Vulnerability Experiment. And so NASA was looking for their next campaign. And they allowed 2 groups to move forward with a scoping study to be able to say, what are the big questions? What should a group be doing? And what ecosystem type should we focus on? One of those groups was tropical rainforests with the Pangea group. The other was a dryland, for the first time, a dryland-focused terrestrial ecology field campaign called ARID, Adaptation and Response in Drylands. So NASA put out the call for scoping study. It picked ARID and Pangea. We had a scoping study that occurred. Many of you were involved, and it was community-based. The community from around the world informed the questions, the themes, the approach. And it wasn't just a research community, it was a decision maker community. So land managers, land management agencies from our country and other countries informed this approach. Both of the terrestrial ecology field campaign scoping studies were so awesome that NASA decided to fund both. So ARID is moving forward, Pangea is moving forward, and now we have, dependent on funding, we have moving forward with a decade of big science. And there'll be an ebb and flow with ARID and Pangea. One project will be funded more while the other is gearing up. Another will come while they're analyzing data. So this is an opportunity because the way that the NASA funding will play out is through funding calls, RFPs that all of you can apply for. We don't get the funding. It's going to be science that can be proposed that can use this cross-scale approach to better understand dryland ecosystems. And so the logic is that dryland ecosystems are poorly understood but globally critical. We have so many decision makers in drylands who need improved information about how to manage these systems in the face of change. Remote sensing tools and ground-based tools are ready in a way they never have been before to capture the significant spatial and temporal variability of drylands. So we can really do things that we couldn't have done before to understand these ecosystems, to see how they're changing and to figure out why they're changing, and to evaluate, consider, try lots of different ways to manage these systems, even in the face of the changes that we now are. Drylands are also a really cool remote sensing test bed for future remote sensing, which the USGS and NASA and other groups care about. How can we use them to understand what we should be assessing next? And so for this experiment, we will blend the satellite, aircraft, drone, a focus on super sites that include eddy covariance towers that give us high temporal resolution assessments of change, carbon, thinking at the plot scale, thinking about the experiments that I like to do. ARID was co-produced with lots of end users, more than we could fit on the slide. I do turn your attention to you and you. and the leadership that CSU showed in determining questions, thinking about themes. There was an enormous amount of engagement. This is over 200 different events, roundtables, community engagement, discussions, not to mention the thousands of meetings we had as a leadership team on top of all of this. We really worked hard to include the expertise and perspective and data needs of wide groups of people. And so from all of you, we came up with these themes, thinking about drought and climate variability, extreme events, the role of water, land-atmosphere interactions and fire, thinking about the structure and function and biodiversity of dryland ecosystems, how they are regulating drylands, how they are changing in drylands, thinking about carbon stocks and carbon fluxes, that variability, and a big focus on social ecological systems, because drylands are social ecological systems. We use them, we live in them, we rely on them. A big focus will be in the western United States. We have tons of awesome drylands and decision makers who are in need of high resolution data, new ways to think about these ecosystems with really exciting opportunities for impact. Thinking about the Bureau of Land Management, for example, it's almost 10% of our national land area, a dominant regulator of dryland systems. If we can give them better understanding and information, they could have national level changes to how they support these ecosystems and the many uses that are in their mission. But also having international work in Mexico, in South America, Africa, and Australia. So there'll be lots of opportunity to be able to do science across the globe. So I hope that you, if you haven't been involved already, you will be involved. Some of you have, and we hope you'll continue. We'll make you continue. Others will force you to be involved who haven't been involved. But you can reach out to us at any time to ask more questions, check out the ARID website. And so I think my take-home messages are like, you guys are rocking science. You are leaders and creating leaders in this field, and it's so delightful and impressive to come and be around you and learn about what you're doing and be collaborating with you. Working with resource managers is awesome. If you're not already doing it, I highly recommend you do it. You can reach out to us at USGS or others if you want some tips on how to go about it. But having your science used in decision making is pretty much the awesomest thing ever. So I highly recommend it. Drylands are super important and responding significantly to change, but their dynamics are not only a challenge to understanding them or to managing them, they're an opportunity. If we can figure out the keys that unlock what's driving them, we could have huge positive impact on managing them. Thinking about super blooms, for example, right? If we can figure out what makes that happen, it's not just moisture and amount, it's timing, it's the seeds being there, it's nutrients in the soil. We can better manage this ecosystem. And now is the time we should do it. We have all the right tools. We have the best community in the world. We have ecosystems that are so important and understudied. And so with that, I will say thank you very much for listening. I love dryland stickers, and I have ARID stickers. So come up and get some swag at your leisure.
I'm sure take a few questions. She did a great job of curbing her enthusiasm. So please ask the question.
I'll start.
Yes.
You know, when I look at the temperature change in drylands, a lot of it happens because they're dry. There's no water to evaporate, the air temperature goes up. And then it's kind of hard to do plot scale experiments because you add water and the surrounding area doesn't have the water, so it just evaporates it away. How do you think about connecting these plot scale water experiments or taking advantage of other gradient experiments to understand the way the drylands are responding to climate change?
Yeah, absolutely. That's an excellent question, and I think the way you kind of answered at the end is I like that too, that we need to pair different approaches to better understand. And so those plot scale give us some opportunity.